The creation of form or morphogenesis in embryonic development is characterized by extensive cell movements and cellular rearrangements. There are two main types of cells in embryos. These are epithelial cells and mesenchymal cells. Epithelial cells are those that are tightly connected, either in sheets or tubes. In these diagrams, the epithelial cells are shown in gray. An example of an epithelium is the epidermis of the skin. Mesenchymal cells, on the other hand, are either unconnected or loosely connected, and because of this, they operate as independent units. An example of a mesenchymal population is the dermis of the skin, and that's the connective tissue that's located under the epidermis. Different cellular processes that occur within epithelial and mesenchymal arrangements guide the process of morphogenesis. Among the factors that guide the process of morphogenesis are the direction and the number of cell divisions. The diversity in the anatomical structures of organisms, even within the same species, is determined by the patterns of cell division. For example, different breeds of dogs have different faces. So even though their faces are made of the same cell types, the orientation of the cells and the number of cell divisions that the cells underwent is different and results in a slightly different pattern. Another example of this is the length of the legs in these dogs. For example, the skeleton-forming cells in the dachshund undergo fewer cell divisions compared to those in the American foxhound, so the dachshund has shorter legs. Changes in cell shape are also required for morphogenesis. For example, the formation of the neural tube, which gives rise to the brain and the spinal cord, involves the formation of a tube from a sheet of epithelial cells. In order for this tube to form, cells need to undergo changes in shape that allow them to form hinges, which are going to be needed for the tube to fold and eventually close. Another process that involves changes in cell shape are epithelial to mesenchymal transitions, and this is where epithelial cells lose their adhesion for their neighbors and they become mesenchymal. Now, these mesenchymal cells have the ability to migrate, and cell migration is also critical for morphogenesis. For example, there is a group of cells in the embryo, and this is invertebrate embryos, that are known as the neural crest cells, and these cells arise from the dorsal aspect of the neural tube, so the portion of the neural tube that faces the back of the embryo. These cells delaminate or separate from the neural tube via an epithelial to mesenchymal transition, and then they have to migrate to distant sites in the embryo to give rise to various tissues like cartilage and bone, and neurons and smooth muscle cells. And this is important because some of the cells that are derived from the neural crest cells include bone and cartilage of the face, as well as pigment cells. Because of this, if these cells fail to migrate properly, that can result in a lack of pigmentation, as well as craniofacial defects, as seen in this child. Morphogenesis is also brought about by cell growth or changes to cell size, and this is really apparent in germ cells, so the sperm and the egg. While the sperm eliminates most of its cytoplasm and becomes smaller, the egg conserves and adds cytoplasm and becomes really large. It also divides asymmetrically to give rise to cell bodies, which cannot be fertilized. In addition to cell division and cell growth, apoptosis or cell death is also an essential part of creating form. For example, bats and dogs are not the only animals that show webbing between their digits. Other animals, including humans, also have webbed fingers and toes during embryonic development, and that webbing has to be eliminated by apoptosis prior to birth. Finally, morphogenesis is also brought about by changes in the composition of the cell membrane and by secreted products. An example of this is the migration of some axons to their correct destination. That process is influenced by secreted molecules that serve as chemoattractants for these axons. An example of that is semaphorin. The migration of the precursors of the sperm and the egg, or the primordial germ cells, is another example. These cells have to migrate to the testes and the ovaries during development, And in order for them to do that, they have to move along a matrix of molecules known as the extracellular matrix that is secreted by other cells. If these cells were prevented from binding to these ECM or extracellular matrix proteins, they would not be able to migrate to their correct locations.